All right. Thank you all for joining us this week for the Living Earth Collaborative and Washington University Eco, Evo, Pop Bio seminar. My name is Mike Moore, and I'm one of the Living Earth Collaborative postdoctoral fellows. And this week, I have the distinct pleasure of introducing our speaker, Dr. Michael Landis, who is an assistant professor of biology here at Washington University. Prior to joining us here at WashU, Dr. Landis has taken a very interesting career trajectory that I think is worth noting. Having trained as a classical oboist, he started out pursuing an undergraduate degree in music performance before switching to computer science. Then, following a few years in the private sector, Dr. Landis spent time applying his computational expertise as a volunteer in a few labs in the Bay Area. One of those labs was Dr. John Hulsenbeck at University of California, Berkeley, where Dr. Landis became fascinated by phylogenetics. Dr. Landis subsequently joined that lab for graduate school and eventually went on to earn his PhD there. After finishing his degree, Dr. Landis held postdoctoral positions in the labs of Tracy Heath at Iowa State and Michael Donoghue at Yale. To accompany this sterling academic trajectory, he was awarded the prestigious Donnelly Postdoctoral Environmental Fellowship, as well as a postdoctoral fellowship from the National Science Foundation. Dr. Landis's research is not only rewriting how we understand historical biogeography and changes in biodiversity over deep timescales, but it is also responsible for the development of several novel approaches to improve how we study these processes and patterns as well. For this work, Dr. Landis has published in some of the top journals in the field, including the Proceedings of the National Academy of Sciences, Evolution, and Systematic Biology. We are grateful that Dr. Landis is able to present on some of his research for us here today, and the St. Louis Ecology and Evolution research community is very fortunate to have him in the city. So with that, I'm going to remind all of you in the audience to please leave any questions that you might have for Dr. Landis in the chat section next to the video, and we'll relay them to Dr. Landis at the end of his seminar. After the seminar, I'd also like to remind those of you in the Living Earth Collaborative and WashU community that there will be a reception where you can continue to chat with Dr. Landis, and I hope to see many of you there. And so without any further ado, I'm going to turn it over to you, Dr. Landis. Thank you.
Well, thank you. That was completely humbling. I really appreciate that. And thanks everybody for joining me today. I'm here to share some of my interest in recent research on phylogenetic models of historical biogeography. And it's a topic I've just been fascinated with since I was a little kid, the idea of species living in different parts of the planet. And I think many of you share that interest, and most of human history, whoops, there we go. And it's not just us as scientists that find this interesting. Throughout human history, essentially every culture has documented where species are found, what continents they're found on, and how that variation might relate to us as human beings. And me as an evolutionary biologist, what's really fascinating about this to me today is how do we uncover or reconstruct how species came to be found where they are? And this is the field that's known as historical biogeography, which admittedly is a little bit of a dusty name, but every year there are new discoveries in historical biogeography that for me just keeps me excited about it and coming back week after week. So here's a recent study that came out in 2020, this year, I suppose. It's a long year. In any case, and it concerns a story that many of you are probably familiar with, the dispersal of primates from Africa into South America. And there are two closely related clades here, the New World monkeys in South America and the Old World monkeys in Africa and Asia. And what's strange about this is when you look at their divergence time, which appears to be sometime in the Eocene, around that time, the continents of South America and Africa were separated by the Atlantic Sea. So if this is true, if this date is true, the Eocene age, it means New World monkeys must have dispersed over the Atlantic Ocean to get into South America. And for some people, that's completely unbelievable. Well, it just so happens that this paper in 2020, they discovered some fossil molars in South America. And guess what? They're nested within this clade of African primate lineages. So it seems that these primates dispersed not once from Africa into South America over the Atlantic, but twice. So to me, that's just remarkable. And for some people, monkeys, not so exciting, not too relevant. We're of course in the middle of a very dramatic historical biogeographical play, which is the spread of coronavirus across the continents. This is an animation from nextstrain.org by Trevor Bedford and Nair. And what's really on the left-hand side, you can see these different sequences that are identified or associated with different continents. And on the right-hand side, it basically shows how it's expected that these transmissions of the sequences between continents unfolded. So this is only to say that historical biogeography has a little bit for everybody out there, no matter whether you care about basic research, natural history, or just, public well-being. Now, my interest in historical biogeography pertains mostly to the development of statistical models and methods to infer how species dispersed between regions, how we can reconstruct where they lived in the past, and so forth. And this is largely a computational effort. And my background is in computer science, like Mike mentioned. Now, I'll show you one example of what a typical analysis might look like. This is an example from an analysis I did on the Hawaiian silver swords, which are a pretty remarkable group of angiosperms. The Silver Sword Alliance is endemic to the Hawaiian Islands. And in this paper with Will Freeman and Bruce Baldwin in 2018, we essentially reconstructed that the silver swords have an ancestral region or range in Kauai, the oldest of the modern islands. And not only that, but we could also use the age of those islands to help date the clade. So this is a fairly standard type of historical biogeographical inference, although there are some bells and whistles in there that I'm not going to get into. Questions that I find fascinating and that my lab is beginning to study more and more are questions such as, how do we develop phylogenetic methods that scale for larger data sets? How do we design models that consider paleogeography, like the ages of islands, when inferring phylogeny and biogeography? And third, how do we design biogeographical models that are built upon evidence and theory from ecology? And this third topic is the one I'll really be digging into today. I know the title of the talk is vague. This is what I'll be talking about. Now, if you're a student in the audience and you're interested in this type of question, then just send me an email later and we'll talk about it. I'd love to hear what you think. Like I said, my work is largely computational. All of the work I'm showing you today are phylogenetic inferences done within a large phylogenetic software package I contribute to as a core developer, which is called RevBase. And RevBase is essentially a model building language that you can use to combine different modeling components and then do inference under such models. Most of my time is unfortunately not spent on Hawaii collecting plants, but instead it's spent writing code. Maybe I shouldn't use the word unfortunately. I find it enjoyable. An addendum: if you're a student who would rather spend your time coding than spending time collecting plants on Hawaii, also please contact me. So like I said, what I'll really be talking about is how do we design biogeographical models to take into account ecology and ecological theory and evidence. And the problem I'll be focusing on is this problem of how do we infer what I'm calling phylogenetic biome shifts, or transitions along phylogenetic lineages between distinct habitat types. And the problem here is if you have a given phylogeny, so here I have the same phylogeny on the left as on the right, and I have the same pattern of habitats occupied by those lineages, I can give you two explanations for how those species came to be in those habitats. So what type of model would help us distinguish between those two situations? That's a difficult problem. And most of what I'm going to talk about today is how might you design models that allow you to better distinguish between alternative histories for these biome shifts. To make this all concrete, I'm going to focus on a plant clade called Viburnum. This is a group I was lucky to be introduced to when at Yale with Michael Donoghue. And this work is largely the result of having an entire army of these botanists at my back to help me understand everything that's so fascinating about this group. Now, if you're from Missouri, you might recognize the name Viburnum, because in the southeastern part of Missouri, in the lead belt, there's all the new lead mines essentially since about the 1950s are part of a newly discovered trend called the Viburnum trend. And smack dab in the middle of the Viburnum trend is nothing more, or nothing less rather, than the city or the town of Viburnum. What's Viburnum? Well, Viburnum is an attractive town nestled in the middle of the Mark Twain National Forest of the Ozark Mountains. I recommend you visit it sometime. And despite the fact that this web page's insignia is I'm not a botanist, I think it's an oak leaf. Viburnum is in fact named after some of the plants that are native to Missouri. And in Missouri, there are 4 species of viburnum. And to many, including myself, when I first started to learn about these guys, is that they look quite similar, but you might like to know that dentatum is as distantly related to these other 3 species, essentially as distantly as dogs are related to cats. So we're talking about 50, 60 million years of divergence. So this is a relatively old group with lots of variation. In total, there are something like 165 living species described and sequenced today. Of those species, we have variation in leaf shape that you can see here. The flowers are insect pollinated, predominantly bees and some beetles, I believe. The fruits are fleshy fruits and probably bird dispersed. In habit, these plants are woody in general. Now, oh, one other thing, the plant leaves, these plants are also evergreen and deciduous, variably, depending on where they live and what biomes they're in. Now, viburnum itself, just for the systematists out there, viburnum itself is within the order Dipsacales. And Dipsacales is a group of about 1,000 species that's nested within the campanulids. There are some other genera that you might be familiar with within Dipsacales, including Valeriana. I learned that valerian, its acid gives the name to the amino acid valine through valeric acid. Fascinating fun fact. Lonicera, the honeysuckles, Sambucus, or the elderberries, which you might know from, what is that, Monty Python, I think. And viburnum, of course, in blue. This topology on the left is an angiosperm 353 topology that I'm playing a small role in generating with these collaborators listed in the bottom right. Now, where do these 165 odd viburnum species live? Predominantly in the Northern Hemisphere. There are 6 major regions of endemism for viburnum: Southeast Asia, East Asia, Europe, North America, Mesoamerica, and South America. And each of these pie charts I've scaled it to be roughly proportional to the number of species in each region. And you can see that East Asia has most of the diversity, about, let's call it 90 species. Southeast Asia, North America, and the Mesoamerica, South America, each of those regions might have on the order of 20 species. One other feature you'll notice about this map is that in each pie chart, you have different colors, and those different colors correspond to the biomes in the bottom left corner. And that's really what we'll start to focus on here. The biomes that we're interested in are tropical biomes, warm temperate biomes, cloud forests, and I might have said cold temperate twice. Warm temperate, cloud, and cold temperate, if I made a mistake. But in any case, you'll see that in the Northern Hemisphere, in North America, Europe, and East Asia, you have a relatively even split between these cold and warm temperate lineages. In Meso and South America, it's predominantly cloud forest lineages. In Southeast Asia, that's where you get essentially all of the tropical diversity. So there's clearly some type of regional correlation with what biomes these different viburnum lineages find themselves in. So here are 4 images of the 4 biomes that I was mentioning earlier. With those collaborators I mentioned earlier, the viburnologists, they shared some images of places they've actually collected viburnums from. So we have the tropical, the warm temperate, cloud forest, and cold temperate forests once again. These are biomes, and biomes are essentially discrete, climatologically and ecologically distinct habitat types. And because they're discrete, I need to give you some definition. And the general working definition I'm using for these mesic forest biomes are that tropical biomes have low degrees of seasonality and no freezing, so you never experience below 0 centigrade temperatures. Warm temperate has some seasonality and rare freezing. The cloud forests have some seasonality and rare freezing, just like the warm temperate forests, but they have more cloud coverage, hence the name, and I believe they also have higher fluctuations in daily temperatures. But cloud forests, of all these biomes, they're most similar to warm temperate biomes. And then finally, we have the cold temperate biomes. The cold temperate biomes experience high seasonality and, no surprise, prolonged periods of freezing in the coldest months of the year. with these different biome types, I mentioned earlier that Viburnum has a lot of variability in terms of leaf morphology. And you find that the leaves of different Viburnum lineages in these biomes tend to sort of converge to the leaf types that you might expect to find any angiosperm to have in these biomes. For example, dentate leaves for the colder regions, large entire margin leaves for the more tropical regions, and so forth. This is just to give you some idea of what we're thinking about when thinking about the conditions that Viburnum originated in. Because how we understand the origin of Viburnum in these biomes kind of dictates how we think these other traits might have evolved, right? So in recent years, there have been some studies trying to reconstruct how Viburnum evolved and originated in these different biomes. And the first paper I'll turn your attention to is this one in 2015 by Beth Briggs and others. And in this paper, they reconstructed a tropical origin for the clade and dubbed this the dying embers hypothesis. And they did so because they found that you had many old but depauperate lineages with tropical characters that informed this root state, yet you had many repeated rounds of more temperate diversification after the early Miocene in, say, 23 million years ago. So this dying embers is to sort of bring to the imagination the ideas of these smoldering tropical lineages kind of burning off into nothing. Now, a year later, there was another paper reconstructing the ancestral biome of the group, and they recovered a freezing ancestry. And they were quite pleased with this result because it conformed with features of wood anatomy. And I, again, have to give the disclaimer, I'm not a botanist, but the anatomical traits they were looking at were these scalariform perforation plates. And my understanding is they sit between the vessel elements and are useful for breaking up air bubbles or embolisms that might cause plants to, die if they experience freezing temperatures and whatnot. But in any case, they found basically the exact opposite result as this dying embers inference. So what's going on here? Did Viburnum originate in the tropical or a freezing biome? We have two stories with two different, almost polar opposites. And what's interesting to this problem about me is both of these papers, generally speaking, we're following standard practices in ancestral state estimation. And the way that ancestral state estimation works is basically as follows. Typically, you start by collecting your DNA sequences and then inferring the species relationships between those sequences using the DNA. And at this point, you don't care about how distantly related the species are in terms of units of geological time. You just want to know how are the species related, the topology. The second step is you then might be lucky enough to have a fossil. And if you have that fossil and it has the right type of morphological characteristics, you can use it to essentially time calibrate or pin down the date of certain splitting events in the phylogeny. And you then use the DNA and molecular clock methods to estimate divergence times. So that's all well and good. Then finally, once you get your time calibrated phylogeny, you can apply a model to infer what these ancestral states are, given the character states that you have at the tip of the tree and the time calibrated phylogeny in the model. You basically rewind time to figure out what happened in the past. Now, what's sort of implicit in this standard practice, and I'm not faulting either of the other papers for following standard practice, but what's implicit in this standard practice is most often after step 2, you essentially discard the fossil. You say there's not much else the fossil can do for me except for give me an age for the clade. Okay. The second thing is the models that we often use to reconstruct ancestral states assume time constancy. They basically say, how you evolved 1 year ago is the same as how you evolved 1,000 years ago, and it's the same as how you evolved 1,000,000 years ago, and so forth. So that's interesting too when you're thinking about lineages and how they adapt to different climates. And these are two very general problems that are encountered in ancestral state estimation, and they're problematic because one, when we do these ancestral state estimates under standard practices, you're normally just using the tips of the tree, extant taxa, to reconstruct these states. And as you go further and further back in time, you expect you have less statistical power to accurately reconstruct these states. The second problem is that over longer and longer time periods, depending on what type of character you're looking at, it may or may not be appropriate to assume the model has the same fundamental behavior over all time scales. And so the question this leads to is, well, how do we get around these two persistent problems? How can we more accurately infer ancestral states deep in time? And this is a metaphor, if I may, of how I view this deep time problem. Imagine you're standing on the precipice of a valley, and you're peering out into the valley, and in the foreground, you can see everything very clearly. This might represent your extant taxa. I can see the grass, I can see the needles on the conifers and whatnot. But as I try to view deeper into the valley, deeper into the past, it becomes more and more obfuscated by this sort of fog of deep time. So in this project with Viburnum and collaborators, I set out to try to find ways to sort of dissipate this fog and make a little progress on the problem. And we came up with two strategies. The first strategy was to try to incorporate information from our fossil taxa that we have for the clade Viburnum. And the second strategy was to try to better design our models so that they don't assume time constancy. And we'll do that by taking information about how we think biomes are structured over time. So the rest of the talk will basically focus on these two strategies and what we learned. So strategy one, infer ancestral biome affinities with Viburnum fossil pollen. We did just that. And the first stage of this was to perform what you might call a modified combined evidence analysis or a biogeographic combined evidence analysis. And what I mean by that is we pooled all the information we had for very different types of data sets: genomic data, plastid sequences, geographical distributions, biome affinities, and the pollen that we could get our hands on, or at least get the data for, I should say. The second stage of this was to fit a joint Bayesian model using RevBayes to all of our combined evidence. And then finally, to measure the effect of including fossil pollen, which turned out to contain some very valuable information for our ancestral biome estimates. What do I mean when I'm talking about combined evidence? Here's an infogram that might give you some flavor. So here I have 4 extant taxa, 1, 2, 3, 4, and my fossil taxon. Now, extant taxa give me DNA. Fossil taxa do not, unfortunately. I wish they did, but they don't. However, all of my extant taxa and even my fossil taxa give me some information about biogeography. And this is what's called a combined evidence analysis. The tip dating part of this name, it comes from the fact that we are going to include fossil taxa as tips in the tree, just like we would include extant taxa in the tree as tips. Now, what hasn't really been done before is people haven't really used fossil taxa while including biogeographical information for tip dating analyses before. So we thought, let's give it a try and see what happens. And one of the major challenges we faced was how to actually code biome data for these fossils, because you look at a fossil pollen and it doesn't necessarily tell you anything about what climate it had or what climate it might have been adapted to. And what I'll show you first, that we were able to assign biome states to these different fossils, and we did it, I think, very conservatively. But I'll show you just one example of what it took for us to do so. So here's one fossil pollen from the Princeton chert in British Columbia, which is in North America. It was from a fossil assemblage about 48 million years old. And that's the early Eocene. At that time, the paleo latitude was about 60 degrees north. We looked at various reconstructions of temperature for that place and time and found that the cold months averaged anywhere between 3.5 to 5.8 degrees centigrade, so warmer than freezing. Also, interestingly, when you look at other fossil taxa found in that assemblage, they're generally frost intolerant taxa like cycads, palms, citrus, banana, and tea. So that all, sorry, my phone's going off. So we basically followed this procedure for all of our fossils and felt comfortable with our assignments. One challenge you face with fossil pollen, however, is that they have very, very few molecular characters. Fossil pollen in Viburnum is not really an exception. And we are lucky, though, that Viburnum seems to have some variation in a particular character called the exine, which is sort of the outer membrane of a pollen. And previous studies have asserted that there seems to be directional evolution in terms of how this pollen exine has changed over time. And I won't get into the details, but this allowed us to at least impose some constraints in terms of where these fossil taxa should belong in our phylogeny. So that's kind of what parts of our data set look like. Let's turn to the model for a moment. Our analysis was a joint analysis, and in Bayesian analyses, you can essentially extract samples from what's called a Bayesian posterior. And we're going to extract samples that include divergence times, species relationships, ancestral range evolution, and ancestral biome evolution. So this is what one of these histories might look like. On the left, we see how it dispersed over areas. Don't worry about the details yet. And on the right, we can see how it shifted between biomes. Like I said, this is one sample, but we can actually generate thousands of samples under this distribution. So this is essentially a way of viewing all the possible ways that we might give rise to the species we see with it, where they're found and what biomes they have, including our fossil taxa. Now, we can condense these Bayesian posteriors into a more traditional format like an ancestral state estimate. Here we found that including the fossil taxa with their range data helped place 2 fossil taxa in North America in yellow here along this lineage, which gives rise to one of the New World radiations. The warm temperate characters we assigned to many of the older biome fossils helped us inform very confidently, at least statistically speaking, a warm temperate ancestry. Now, what would happen if we basically erased the biome information we had for the fossil pollen? When we do that, we get this very ambiguous result about what happened at the time of the origin of Viburnum. It's about 50% warm temperate, 25% tropical, 25% cold temperate. That for me is not really enough to really say with certainty we know what the ancestral biome is. Now I'm going to come back to this point in a second, but we can also summarize our posterior distribution in another way. We could, for example, ask what biomes did Viburnum tend to occupy when they were in North America? So we could ask for a particular region, what is the proportion of different Viburnum lineages in each of the biomes that we're interested in? And we found that early on, in general, we have large representation in warm temperate biomes. And not until we hit the Miocene at about 20 million years do we see a rise in the more cold temperate lineages. We can do this for all 6 of our regions, of course. And we found low persistent diversity in tropical forests in Southeast Asia. In all of our northern regions, we have this rise of cold temperate biomes in those 3 regions. And in sort of Meso and South America, we have predominantly cold adapted lineages. We can invert the problem. Now let's ask, for each biome, what regions do you find those lineages in? So here's one looking at the warm temperate biome. And we found that early on, most lineages were probably in East Asia if they were warm temperate adapted. Our fossils give us this nice little peek showing that there was, in fact, among warm temperate lineages, a little bit of a peak in terms of North American diversity with respect to other regions. And again, we can do this for all of our 4 biomes. And we find that tropical diversity rose faster in Southeast Asia than East Asia over time. And that in the cloud forests, we have this sort of succession of North American, then Central American, then South American colonization. And we can look at all of these together, and you see something like this. Now, I only want to show you this to contrast it with what these lineage proportion through time plots look like when we remove our fossil evidence. Now we have an uptick in cold temperate lineages in Southeast Asia, East Asia, Europe, North America, you name it. And we lose our peak of warm temperate representation in North America, which would be the yellow color in this panel when we ignore the states of these fossil taxa. One thing you might ask if you're familiar with the fossil record is you probably notice these fossils we have are all warm temperate. They're all in Europe and North America, and there are various reasons to be concerned with that. One is taphonomic bias. It might be that maybe warm temperate biomes just preserve fossils better than other biomes. That's possible. It could also be a scientific bias, being that some regions of the world are harder to do field work, but also that some regions of the world where local scientists are reporting the fossil record there are less cited in, say, Western journals. And so that would obviously bias our results, and not on top of being unfortunate in general for scientific discovery, right? So one thing we did to get a sense of how our reconstructions looked and how sensitive our results would be to these fossil taxa and potential biases was perform a sensitivity analysis. And I won't get into these details, but we estimated that we would need to discover something like 12 new fossils that were exclusively cold temperate and of the similar ages, say in the Eocene, in order to flip our result from a warm temperate to a cold temperate result. So that would be surprising. And it would be, whoops, wrong direction. It might be surprising because the distribution of biomes over time has changed an incredible amount. And where we're looking in the Eocene is about here in this sort of light peach color, if you can see my mouse. At that time, there were some cool forests, but essentially no cold forests anywhere on the planet. And when we look at the Landis et al. Estimate, which is the freezing temperate estimate, which is about 55 million years old for Viburnum, that's basically right at the peak of the Paleocene-Eocene thermal maximum, so very warm time on the planet. And I'd be surprised to find cold temperate fossils during this period of time. Now, when we get further towards the present, the world is cooling, it's getting drier, and that's when we start to see the rise of these cold temperate forests. And this realization led to a second idea, which is the second part of this talk. How might we infer ancestral biome affinities using paleobiome structure? And the basic idea here is that we're going to design a model where paleobiome structure informs the probability of these histories. We'll fit it to the Viburnum data set again, this time not using fossil taxa. And then we'll again compare how our results differ based on including or excluding these fossil taxa. So let's see what happens. The first step is we need a model, I suppose, if we're going to model something. So how would we define this model? There are two major components or challenges we face here. One is, how do we model the biome structure and how it might change over time? So that's one problem. But secondly, how would we model the way that phylogenetic lineages would shift between biomes over time? So two related but distinct problems. Let's look at the first one. For the first one, we address this issue of how to model biome structure is by saying that each region can contain some number of biomes. And then secondly, we might say that the regional biome structure itself can vary over time. So these are the two sort of guiding principles in what I'm about to show you. Let's look at present day structure of tropical biomes. So I've penciled in where tropical biomes generally are regarded as being today. And you can see that they're highly disconnected, largely centered around the equator. What I can do is I can take that structure and encode it in what I'm calling an adjacency matrix. And this adjacency matrix has two major features. One is that along the diagonals, I am representing whether or not a particular biome is present in a particular region. And on the off diagonals, I'm representing whether or not this biome has connectivity between those two regions. And I can add features that describe whether or not this connectivity or this presence of the biome is marginal, weak, or strong. So that I can relax some of the assumptions about how exactly I have to define this structure with respect to different time periods and different biomes. Now let's look at a different time period going back to the mid and late Eocene. Well, tropical forests certainly were more expansive then. And we might expect to see that reflected in these adjacency matrices. So now we see presence of tropical forests in all of the regions that I defined, which are the same 6 regions as in the first study. And we have more off-diagonal cells filled in red, meaning there's more connectivity between between regions through the tropical biome. That's the main thing I want to drive home here. And I can define this not just for one time period, not just for one biome, but I can do it for, in this case, 8 different time periods, all 6 regions and 3 different biomes. So the tropical biome, the warm temperate biome, and the cold temperate biome. I'll mention here that I've collapsed the cloud forest and warm temperate biomes together for various reasons. And constructing these matrices was not easy, and it is somewhat subjective, but I stand by it. It's built on references, qualitative descriptions for various types of paleobiome structures, like the one that I showed you a few slides ago. Now, there are two major features that really pop out here. One is that before 34 million years ago, before the onset of the Oligocene, the structure is dominated by these tropical and warm forests, and you have high degrees of connectivity between them. But you notice there's an absence of these cold temperate forests, however. And when I talk to experts in this field, they really say it's true. There are very few cold temperate forests around at that time. So in this first time period, you have sort of the reign of the boreotropics. In the second time period, you have the reign of the boreal forest, which is, these are the northern forests we're used to seeing today in Canada, in Russia, and so forth. Tropical forests become, they start to be separated, or rather they fragment and become disconnected. Warm temperate forests still hang around, but it's the cold temperate forests that really rise to dominance and become connected throughout the northern hemisphere. Okay. Whew. So now let's look at the second part of the model here. Next, we need to ask, how can we actually model the way that lineages would shift between regions and biomes? Well, first, what I'm going to do is I'm going to say that lineages have not a separate biome state and a separate region associated with it, but we have a compound state that is sort of all the possible combinations of biomes and regions for each lineage. So we're now going to talk about a lineage being in both North America and in a warm temperate biome simultaneously. Next, we're going to define a model that defines rates for how you transition between these biome region states, these compound states. And then finally, we want to define the event rates such that they depend on the paleobiome structure I just defined in that first part. So those are the three steps for the second bit. So here I've just basically listed out the 6 times 3 biomes that would give rise to the 18 possible compound states. And I've color coded them up here. Pink is Southeast Asia, red is East Asia, and so forth. If it's a light color, it means it's tropical. And if it's medium, it's warm. If it's dark, it's cold. And so this is the general schematic I'll be using for representing these compound states. Let's now try to evolve a compound state. How might that look? Whoops. Right, okay, so how might that look? What I'm going to do is I'm going to define a rate matrix over the 18 possible states. And this rate matrix will have a very particular block matrix structure. And what I mean by that, and you can see how I've color coded it, is that events within different cells of this block matrix structure will relate to specific types of evolutionary events. So if you're on the diagonal blocks, it means you're dispersing between regions. If you're on the off diagonals in blue, it means that you're shifting between biomes. And I've made an assumption in this model that you can't shift biomes immediately between tropical and cold temperate forests. That's an assumption you could change if you wanted to, but I'm comfortable with that. So in any case, let's take this matrix structure and try to evolve a lineage. How would that look? So suppose you start in the states warm temperate and East Asia. So this is the medium red color. And this is a stochastic model, meaning things happen at some random time, and what happens is also random. And we're trying to infer what happened in the past through this stochastic model. So some amount of time happened, or elapsed, I should say, not happened. And during this time interval, when we reach the end of this time interval, rather, something happens. What happens, we don't know yet, and there are different things that could happen. It could be that we shift biomes by selecting one of these blue elements on our rate matrix, or it could be that you disperse between regions by remaining in the central block diagonal matrix, the red one. So what do you think happened? I guess here I decided that we had a dispersal event. And so the next event drawn randomly chose a dispersal event into the North American region. Like, note that the biome did not change. So it was warm East Asian and now it's warm North American. Let's let some more time elapse. And then something else happens. What could have happened? Here I decided to have a biome shift. So now, instead of dispersing into another red colored cell, we shift into a blue colored cell. So we shift from warm temperate into cold temperate. And that just goes on and on and on. So here's the next tricky part, is we want to somehow define these biome shift and dispersal rates, the blue and the red cells in that matrix there, to somehow vary or depend on the time of the process, the region a lineage is in, and the biome the lineage is in. So there's at least 3 pieces of information we have to keep track of as we're computing these probabilities. And so I haven't clicked next yet. That's what I just said. So we're gonna somehow define these rates in terms of as functions of time, region, and biome for a particular lineage. And here's an example of what a rate matrix structure might look like if we were viewing the mid to late Eocene. And I'll show you a little bit more about how this looks in a moment. Now, one challenge here is that for any given species or any given clade really, you don't know a priori how important biomes are to their dispersal abilities, right? So it could be that a species is an immaculate generalist and it can disperse between regions no matter what biomes are present. And if that were true, you might expect a lineage to disperse something like this. And so this is a process I'm calling the Null process. And it says that in effect, all biomes are present in all regions at all times. It's a model, and it's quite similar to models that are used to typically estimate ancestral states. And what you'll see here is at the very top of the screen above the matrix, time is ticking by. And what this says is for each of these blocks, for each of these matrices, nothing is changing the rate matrix structure. You don't see any changes here. Biome shift rates are constant and dispersal rates are constant. Now let's consider a model where terrestrial connectivity matters, but biomes don't matter. And the biome shift rates then are constant. Dispersal rates vary with time and region. And one thing, if you're a fan of Cenozoic paleogeography, is you'll know that for these 6 regions that I'm talking about, not much changes in terms of their continental configurations with respect to one another. The most exciting event, I would say, is probably the closure of the Isthmus of Panama. And you'll see that even as time ticks by, my rate matrix remains fairly constant. So not much exciting is happening there, but some dispersal is limited. Finally, let's look at paleobiomes. So this is what I'm calling the biome-aware process. Here I'm using the 3 paleobiome, the full paleobiome structure time series for the 3 biomes and 6 regions I described earlier. And you can see here something interesting is happening. We're seeing dispersal rates vary depending on what biome you're in, what block row you're in. We're seeing that biome shift rates depend on what region you're in. So the diagonals of the off-block diagonals, that's a mouthful. So like here you see that depending on what region you're in, it's easier or more difficult to shift into a tropical biome from a warm one, and so on. And this is, in my opinion, probably what is most reflective of how lineages shift between biomes and disperse. But no reason to take my word for it. We can try to estimate which of these 3 processes is sort of the dominant one when describing variation in regions and biomes. And so, not to get into too many details, we set this model up such that it's a mixture of models, and we can infer the mixture weights corresponding to these 3 models, these 3 processes I just introduced, the Null process, the geography process, and the biome process, and we'll infer how important the weights are, each process is, from the data we have in our hand. So if the biome shift, the biome structure model really matters in the bottom left, we should infer a large WB parameter, the weight parameter. So let's get back to Viburnum for a moment. So I analyzed the Viburnum dataset again using essentially the same species. I removed some species that had low support in terms of their placement, so it's slightly smaller. I used the 3 biomes. Remember, I collapsed the cloud forest and warm temperate forest into 1 biome, and the 6 regions I described earlier. Here's the familiar paleobiome structure configuration I mentioned earlier. And what I'm going to do now is I'm going to analyze Viburnum using 3 different versions of the biome structure model on the right. And the first thing I'm going to do is ignore regional and biome structure altogether. So I'm going to assume that all lineages evolve under this Null model, no matter how old they are, no matter what biome they're in, no matter where they live on the planet, they always evolve under this black adjacency matrix. The next thing I'll do is I'll misrepresent the past and assume that biome structure has always looked like it does today. for the past 100 million years. And then finally, I'll use the paleobiome model, the paleobiome structure model, where I'm using the full set of information that I've collected here. And let's see what happens when we fit these three models to Viburnum. Well, first, we can fit it to the paleobiome data set. We get this medium red color, which is warm East Asian. We can fit it to the modern biome structure, where we get this dark red color in high probability, which is the cold East Asian origin. And then the null biome, where we ignore biome structure altogether, and we get a more even split between warm and cold temperate biomes, but still in East Asia. Well, that's interesting, isn't it? So we're fairly confident about where Viburnum originated, East Asia. But depending on which model you assume for the structure of biomes, we get three fairly inconsistent results, with the paleobiome model favoring warm and tropical origins, and the modern and null biomes favoring cold and warm origins. So that's a little concerning. One thing you can do, though, is remember I said we can fit these mixture weight parameters to the data set and ask what parameters did we actually estimate? And using a little Bayesian model testing magic, we can essentially rule out that the null model is supported. When we fit the paleobiome model and the modern biome model, we get very strong support that biome structure matters. However, we get two completely different results in terms of what the ancestral biome is. That's pretty interesting. And by the way, if you compare paleobiome to modern biome, paleobiome gets a better score. But what's concerning here is that if you fit your data set to a modern biome structure, you might think that biome structure matters a hell of a lot, but it's actually not the correct answer, potentially. There's just three last slides for this part of the talk. And I want to show you slides that were analogous to the end of the first strategy I used, these lineages through time. And if we look at these lineage through time plots, the lineage proportion through time plots rather, we see that we have a strong dominant component of warm East Asian ancestry, all the way up into the Oligocene, when we think these cold temperate forests first really originated. For the modern biome analysis, we have more cold temperate than warm temperate lineages in East Asia. Okay. And it looks similar for the null biome. If you compare these trajectories to the results I got in the first analysis, they map on quite nicely. The paleobiome result is almost identical to what I got for the reconstruction for strategy 1 when I use fossil biome states. The modern and null inferences are almost identical to what I got when I excluded those fossil taxon biome states. So now I have two sort of independent ways of trying to reconstruct ancestral biomes, and they're using different lines of evidence to reconstruct the past, but they seem highly concordant. And what can I say? I guess maybe I like to compromise, but when I was posed with this question, I came at a different result than I think some of the collaborators were expecting or hoping for. I think it's a warm temperate origin, not tropical or freezing personally. And it's because I think it's largely driven by, on one hand, we have so many warm temperate lineages alive today. And simply, we don't have these freezing forests long ago in the past. On the other hand, our fossil taxa for Viburnum really seem to be warm temperate fossils. They don't seem to be tropical or freezing ones. But it'd be interesting to see if that story changes with more discoveries. So I'm going to recap everything I shared. I introduced two inference strategies. The first one was to perform a combined evidence approach, focusing on biogeography and fossil taxa. The second strategy was to design this time-dependent model where we infer biome shifts within this paleobiome structure. One thing I'd like to mention is there's something interesting about these studies on small clades that I didn't previously appreciate, and it's that there's something to be said about very careful analyses on smaller clades where you really know the data set and know all the species. And this is not at all to slight the Zan et al. Paper, which represents a large angiosperm reconstruction, and I like the paper very much. But when you work at these large scales, these big trees, at some point, when you start to work with tens of thousands of tips, parts of the reconstruction might get away from you, and you might not know exactly what's going on. And as much as you can learn from these big tree analyses, there's still something to be said about these focused studies on smaller clades. And I think we'll start to see more reciprocity between the two, where small clades, where you really work out the details, can be used to help sort of check, or I come from computer science, but unit test. Here I'm calling it a canary, to sort of flag where you think the result might be correct, or maybe a place where your your big tree analysis is a little bit off. Another thing I'll mention about these phylogenetic inferences for biome affinities, the biomes are still changing. This is a forecast of how biomes might change by 2100. The main thing I'll point out is, you probably are aware, there's likely to be a loss of ice, tundra, and rainforest. We're in the midst of it now. I mean, the Amazon's burning, not to be a downer. These phylogenetic estimates, however, potentially could help us understand how tolerant different clades are of adapting to changes. I mean, if we find that some clades are perfectly happy shifting into new biomes when it arises, well, maybe they're not at such a high risk for extinction. Other clades, if it seems like they've never gone between, if they've never left the tropical biome, that could be quite frightening. I'll move on to something lighter. A pet interest of mine is understanding how paleogeography can help date phylogenies. And I think there's a lot of room to incorporate information about paleobiome structure to help estimate the ages of different groups. The example I showed right at the beginning of the talk on the silver swords was ignoring climate, was ignoring biome structure and so forth. But let me tell you, the silver swords are nested within a very arid adapted clade called the tarweeds in Western North America. And some of our most beloved silver sword, members of the Silver Sword Alliance, the silver swords themselves, for example, they're found in these sort of alpine deserts on the highest islands and the youngest islands. And so it's likely that certain species might have an easier time colonizing, say volcanic island systems, depending on the age of the island itself, because these high altitude alpine deserts that you find in Hawaii are on after periods of tremendous uplift from volcanism. Thanks for indulging me on that. The way I'm sort of viewing fossil structure or fossil data and paleo data in general these days is, it's sort of our only portal or view. I think of it sort of as a beacon in the fog to see past and penetrate this deep fog, to sort of clear it up. And I'm not the first person to think this by any means. I've really been inspired by papers that have come out over the past decade that are very carefully trying to take concepts in methods development, statistical models, what we know from molecular evolution, what we know from the fossil record, and unify that. And I really feel like this is our road forward to getting a comprehensive view of evolutionary biology for these deep timescales. We're not going to learn about biome shifts just by looking at DNA sequences, I'm sorry. But it's also likely that we're not going to learn about how, say, a group like Viburnum evolved if we only have 5 fossil taxa. And to abuse a quote by Charles Lyell to the point, I guess, of tautology, I think that many of us in the phylogenetics method community are recognizing that, the past is the key to the past. Why are we inferring everything? The data is there. We need to find ways to bridge these different data sets, these different viewpoints and whatnot. And what I'm going to end basically by saying, if you work on methods, I really implore you to work closely with empirical biologists. I did that during my postdoc. It was probably one of the best things I've done in my scientific career. If you work on empirical biology problems and you see something wrong with a method, definitely reach out to that methods person and help educate them, because there's so much room to there's so much work left to do to get all these models working correctly. If you're reading this list carefully, please don't feel offended if your name's omitted. There's too many wonderful papers coming out to list them all. With that, I'd like to thank you. This is sort of strange because it's a YouTube talk, but at the same time, I'm mostly addressing people from WashU. You might know that I had a daughter, Annika, maybe 8 months ago. She's been fun to hang out with. But I just wanted to share that everything here is going fine. If you want to talk about babies, I would love it. If you want to talk about biogeography, I would also love that. Or if you just want to say hi, I hope we have the chance to do so. So that's all I have to say. Thank you very much.
All right, thank you, Michael. That was a terrific talk. I'm sure that everybody at home is thunderously applauding. And so we're gonna go ahead and get the questions, the question and answer period going now. And so just a reminder to the audience to leave any questions that you have now in the chat if you haven't done so already. So I'm gonna start off with a question from Rachel Penchikowski. And she was wondering if dispersal of bird lineages might help with the inference of dispersal of plant lineages like Viburnum. And are there any sort of known specialist bird dispersers of Viburnum species?
For Viburnum, they have these fleshy fruits, drupes, and as far as I'm aware, people think they're bird dispersed. I worked with some of the field biologists who were studying Viburnum, and they said they actually weren't entirely sure exactly what bird species ate the fruits. So that creates a kink there. With other groups though, like the tarweeds, the silver swords, which are in the tarweeds, as the name tarweed suggests, they're pretty tarry, and it's thought that birds probably helped disperse them to the islands long ago. But with many of these relationships between birds and fruits and dispersal syndromes, it's not as tight as you might want it to be. But I really love the idea. I hope we could figure that out someday.
Great, thank you, Michael. Really, really fascinating talk. I'll follow up with another question here. And this relates to something that I found interesting. I think it's so fascinating that fossil pollen structure can be so well preserved across time. A question that came in is, did you say that pollen structure is indicative of the biome of the plant, such as temperature or humidity?
No, and if I said that, I didn't intend to. The fossil pollen structure itself, and in particular the exine, I'm not a palynologist nor a botanist, keep in mind, but as I understand it, that structure is very distinctive for major plant families, and it does preserve well over time. And that's a fundamental source for how people identify what plants were in what places at what time when you look at the fossil record. I'm not aware of fossil pollen structure indicating biome, but it might. I would confer with a palynologist to figure that one out. I could imagine possibly.
Great. So I was sort of curious about if there's a way that you can account in these models for like asymmetries and how difficult you might expect it to be to adapt to different biomes. Like, for example, I could imagine, some of these like really arid or really these like sort of tundra biomes might be more difficult to adapt to than a tropical one, and whether you might be able to account for those kinds of asymmetries.
Yeah, there's some ideas I'm playing around with. And one thing I did in the biome shift model where I was looking at paleobiome structure over time, I essentially forbade lineages from going from tropical to cold temperate forests. And that's basically because in reality, you never find those two biomes adjacent to one another. In Viburnum in particular, there are no lineages where they would plausibly shift directly between those biomes. So it's an assertion you're making about the structure of biomes, and biomes have lots of structure like precipitation, temperature, and whatnot that you could use to sort of encode that structure too, and potentially also influence asymmetries. But yeah, that's worth exploring in the future, absolutely.
Great, and it looks like you've inspired some of us in the audience to do some modeling ourselves. Somebody asked here, in your opinion, what is the best introduction to biostatistics textbook?
I don't know. I really don't know. I think it will depend on your discipline. If you're working in cell biology, that's clearly different than ecology. So I would probably work with people whose quantitative folks or other people learning statistics in your field who you admire and ask them, what can I learn to sort of get to where you are or to follow in your footsteps? My background is in computer science. I was not a great math student, and I learned most of my statistics as a PhD student in a biology lab. And the statistics I learn and I know are probabilistic models and Bayesian inference. That's really my expertise. And that came out of the research, I wanted to learn how to use these statistics for the problem that was in front of me. So I have gaps in my knowledge. And I personally think that research is a great motivator for learning, but for a broader general biology stats textbook, I couldn't recommend one unless I knew more about what their discipline was.
All right, so it looks like it's about 5 o'clock now. So we're gonna go ahead and end the presentation section. And I hope that those of you in the LEC and WashU community will join us over in the reception in just a few minutes. So once again, I'd like to thank Dr. Landis for his really interesting talk today. And I look forward to chatting with him more over in the reception. So thank you all for joining today, and I hope you have a nice week.
Thank you. I really appreciate it.